Good morning, everyone. My name is Yedemo. Today's seminar is based on the publication of Hu and collaborators this year entitled Spermidine Enhanced the Antioxidant Capacity of Rice Seeds During Seed Aging. We will follow this presentation plan as shown on this page. First of all, let us discuss about the importance of seed viability. As you know, the long-term seed conservation is important for food security. The seed survival during long-term storage is important for the conservation of valuable genetic resources. The frequency and uniformity of seed germination are important for crop yield and quality and has high economic relevance. It's also known that high temperature, high humidity, high oxygen vapor pressure, and long storage intervals can reduce the germination of seeds. So the seed viability decreases as they undergo aging, leading to severe economic losses during seed marketing and loss of germplasm resources. Today, our presentation is based on rice. And as you know, rice is an important food in the world. And the study on the regulation of seed deterioration and the maintenance of seed viability may contribute to the production and the storage of rice seed and also the effective management and conservation strategies for seed under long-term storage that has a very great biological value as well as economic significance. So today, what do we know before this study? What was the previous work uh, before the present study? So many studies have investigated the biochemical processes and internal mechanism of seed deterioration and changes in vigors during storage. Seed aging has been suggested to be associated with the expression of DNA and protein repair genes, telomere length, epigenetic regulation of DNA methylation, and changes in organelles and nuclear genomes. Aging is also manifested as the accumulation of reactive oxygen species, mitochondrial damage, changes in the antioxidant system, and lipid peroxidation. So higher temperature, humidities, and oxygen concentration promotes the formation of reactive oxygen species in the seeds that lead to the damage to macromolecules, including protein, lipids, nucleic acid, and as a result, the destruction of mitochondria and affect the normal repair of the morphological structure. So, to deal with oxidative damage caused by high concentration of reactive oxygen species, plant has involved a complex reactive oxygen species mechanism to maintain balance that include some enzyme like superoxide dismutase, ascorbate peroxidase, catalase peroxidase, and so on. So for today's presentation, why the authors are interested specifically to spermidine? Why the choice of the spermidine product? Well, in the literature, spermidine is one member of polyamine, and spermidine is present in almost all living organisms, and his function is related to modulation. For example, in a transgenic European pear, the overexpression of spermidine has been shown to be associated with strong resistance to various stresses. Another example is during seed germination of sweet corn, spermidine was demonstrated to promote fast seed germination and high seed vigor, as well as potentially playing an important role in maintaining cell membrane integrity. So polyamines, including spermidine, have been suggested to initiate plant defense systems and improve plant adaptability to various stress. However, there is a little information regarding the role of spermidine in the case of rice. Therefore, the present study used spermidine to test on a Chinese cultivar under aging pressure. So in this case, what were the scientific questions that the authors want to answer? First question is, what is the spermidine effect on rice seed aging? Second is, what is the effect of this product on rice seed germination? Third question, what is the phytohormone content in rice seed during the spermidine treatment? Fourth, what is the antioxidant activity? And five, which genes are involved during this process? To answer those questions, the authors define a set of methods. Firstly, they take mature seeds and soak in water or spermidine for 12 hours. And then the pre-treated seeds were hair dried at 28 degrees C for at least 3 days. The seeds were then placed in an aging box, that is Thermo Fisher Scientific Incubator Model 3111 from USA, at 43 degrees for 4 days with a high relative humidity of 98%. And here we have three main treatment. SBA here is a spermidine treatment coupled with aging pressure. WBA is water treatment coupled with aging pressure, and CK is just seed with no aging pressure. So the next step is to test the germination of the seed and the growth during the process. So for this work, the authors placed the seeds in a germination chamber with 8 hours of light and 16 hours of darkness. And during the light time period, the temperature was 30 degrees, and during the darkness period, the temperature was 20 degrees for a total number of 14 days. They set 3 replicates. Each replicate is composed of 50 seeds. And then the seed germination was measured using some parameters, including the germination energy, the mean germination time, the germination index, the germination percentage. And then the mean germination time and the germination index was calculated as described on the page, with GT as the number of germinated seeds at DT, and DT is the time corresponding to the record date since the beginning of the inhibition. Also, some plant hormone content were measured. So the author measured extract and purify abscisic acid, ABA, and gibberellic acid, GA. Furthermore, they use ELISA to perform the hormone content in the seed sample. In addition, some enzyme activity were also measured. So the catalase activity, the superoxide dismutase activity, the ascorbate peroxidase activity, and the MDA content, malondianate, was also measured. And then they also used to measure some amylase activity, alpha amylase and beta amylase, as well as peroxide, hydrogen peroxide activity also was measured. And then the data, all collected data, were analyzed in SAS software using analysis of variance, followed by mean comparison using the least significant difference test. So after this set of methodologies, what are our results? So as a result, globally, the aging pressure significantly decreased the rice seed germination from 0.86, that is the control, to 0.42 here in the water treatment. Moreover, the spermidine treatment had a higher vigor and better seedling growth than those of water treating. So you can observe here that the spermidine treatment has higher germination index, germination energy, as well as a higher germination index. So the spermidine seems to be a good alternative to restore the seed germination during an aging process. This figure shows the effect of the spermidine on the hormone, precisely gibberellic acid and then abscisic acid. So for gibberellic acid, you can see here there were no significant differences at time 0 because all value, all mean has the same letter. So there is no significant differences at time 0 among the three treatment. But at the same time 0, there was a significant difference for ABA contents following the three treatments. Interestingly, there was no significant difference of abscisic acid at 12 hours after imbibition. You can see here, but the ABA content was higher under spermidine treatment than water treatment. So the ABA levels has been decreased from 0 to 12 hours after imbibition in control treatment. This observation have been previously reported in Arabidopsis, but however, uh, the level of action of the spermidine, the role of this product in the germination during the aging process is still uncovered. So further investigation is required. And then we also noticed that the results of gibberellic acid have been increased under spermidine treatment. So as you know, the gibberellic acid is known as hormone that guide, that promote the seed germination. So this study showed that the spermidine could help to improve the seed germination under aging pressure. So this next figure shows some enzyme and as alpha amylase and beta amylase, as well as hydrogen peroxide concentration for the tree treatment. I mean, CK, that means the control, WBA, that is the water treatment, and SBA, that is the pyrimidine treatment. So for hydrogen peroxide content, the level was higher in water treatment than the two others one. Moreover, the activities of alpha amylase among the three treatment here remain at similar level. You can see here, here, and here. So the alpha amylase activity remains similar. However, the beta amylase activity is significantly different among the three treatment. But when we compare water and spermidine treatment, the spermidine treatment has higher beta amylase value. So in this study, the beta amylase activity has been increased under spermidine treatment with the stable hydrogen peroxide content compared to the control. But the beta amylase is known as an indicator of the germination ability and vigor of the seed. So in our case, the spermidine can help to delay the aging process for this specific rice genotype. Therefore, the spermidine has a great effect for cope the aging process adverse effect in the rice based on beta amylase content. The next result is about the antioxidant activity also. In this study, they also try to investigate antioxidant activity, antioxidant system in the rice seeds. So after 14 days, there is no significant difference in activities of catalase here. We can see here, this is the same letter AAA. There is no significant difference at 14 days here, as well as here also for this parameter, that is the superoxide dismutase. In addition, the ascorbate peroxidase, those two parameters, APX and MDA, MDA is malondialdehyde activities, were increased. You can see these activities were increased for water treatment to spermidine treatment, water treatment to spermidine treatment. So at 3 days and at 40 days, 14 days, those rates increased. Those rates increased for these two components. So these results show that the seed quality and the seed activity of some antioxidant enzyme were significantly increased during the aging process. But it is known that the increasing of the antioxidant system can lead to a low loss of the seed viability. So the antioxidant systems was activated here, even though there is the presence of spermidine. The authors also investigate the gene expression of some antioxidant systems by using real-time PCR. So this figure shows the expression of the catalase, superoxide dismutase, ascorbate peroxidase related gene among the different treatment. So you can see here in red, at 12 hours after inhibition, the aging treatment improved the expression of catalase B. You can see here, the red color means the increase and the blue color means decrease. So you can see the CATB, the SOD1, SOD3, as well as APX1 and NPX3 has been increased. But compared to the 3 days, the activities of those enzymes generally was low, except for SOD3 and APX2 and APX3 that increased. At 14 days, at 14 days, we can also see that the expression of TATB was reduced, in contrast with expression of CATC and those others enzyme like APX1, 2, 3, and APX4. So those results was in conformity with our previous result, specifically for the day 3. But there is some limits for these studies, and we can suggest some future direction. The first point of the limit of this study is that the regulatory and signaling roles of the spermidine is not covered here in this study. To cover this point, omics-based analysis is required, as well as high throughput phenotyping. The second point, second limit of this study is that the authors only use one genotype. One cultivar was used for this study. So we need to enlarge the genotype diversity in order to study the diversity reaction in contact with the spermidine. So as a conclusion, we can retain 3 major points. The first is the aging treatment affects rice seed viability and germination. The second point is the spermidine can help to boost the seed germination, the seed vigor. We previously saw that in our previous presentation. And thirdly, there is a need of additional study to find the specific pathway using omics tools and precise phenotyping, as well as more diverse rice phenotype. So we are at the end of our presentation. Thank you very much for your attention, and your question will be welcome. Thank you very much.